Okay, so we stopped here at lecture 4.1. At the end of the first lecture of chapter 4, we stopped here. And I'm starting here in lecture 4.2 because we're going to talk about organelles. We're going to talk about all the different organelles that can be found in the eukaryotic cell. So let's get to it. There are 4 categories of organelles. These are my categories. I'm trying to organize them to make it just easier to lecture to you. You're not going to be held responsible for these 4 categories of organelles because they are my categories. I'm not going to test you on this. I'm not going to ask you the question, please list the 4 categories of organelles. I will not do that. This is simply a way to organize this lecture. I'm going to first talk about the nucleus and ribosomes. Then I'm going to move to the endomembrane system. Then I'm going to go to organelles which relate to energy. And then finally, I'm going to talk about the cytoskeleton. Okay, so we're going to talk about most of the organelles pictured here. Do not worry on a test. I'm not going to make you identify them by the way they look. I'm not going to make you draw anything. I'm going to hold you responsible for their function. So when we go through these organelles, know the function. Okay, so the nucleus and ribosomes. The function of the nucleus is to keep the DNA away from the cytoplasm. There are reactions that occur in the cytoplasm. The nucleus keeps the DNA protected from these potentially damaging reactions. So prokaryotes do not have a nucleus. They don't have this protection, but eukaryotes do. They have this structure called a nucleus. So on the outside of the nucleus, you have a nuclear envelope. You can think of that as the nuclear membrane. It is just like the cell membrane, except it surrounds the nucleus. Okay, it surrounds the nucleus. It is called the nuclear envelope. And like the cell membrane, the nuclear envelope is like the bouncer. It's there to keep stuff out and to keep the DNA in. Okay, so here's a picture of the nucleus. We have the nucleus in the cell. And remember, you have this plasma membrane or cell membrane on the outside. We talked about that in the last lecture. We are now focusing on this organelle called the nucleus. There are 4 parts that you need to know in the nucleus. On the outside of the nucleus, you have the nuclear envelope. It's like the cell membrane of the nucleus. It keeps stuff in and it keeps stuff from going in that nuclear envelope. Kind of like the bouncer in the cell membrane. Inside the nucleus, you have nucleoplasm. Inside the nucleus, you have this structure called the nucleolus. And you also have chromatin. I'm going to go through these 4 parts next. Okay, let me move me. Here we go. Here's me. Okay. So, the nuclear envelope. Again, we are talking about the outside of the nucleus. The nuclear envelope. It's like the cell membrane for the nucleus. It's actually composed of the same molecules. Phospholipids, just like the cell membrane, except it's even thicker than the cell membrane. Remember, it's a lipid bilayer around the cell. Around the nucleus, 2 lipid bilayers, two lipid bilayers, 4 molecules thick, 4 phospholipids thick. So you got 1, 2, 3, 4 phospholipids, 2 lipid bilayers surround the nucleus. That is what composes the nuclear envelope. So again, just to remind you, if it's in red, it's not in your study guide. You're not responsible for it. It's just, ooh, ah, information. Okay? So what you need to know about the nuclear envelope. There are 2 lipid bilayers that surround the nucleus. 4 phospholipids thick. And those phospholipids have the same thing as the phospholipids around the cell membrane. They have the polar heads that like to interact with water and the tails that are hydrophobic that don't like reacting with water. Okay? So 2 lipid bilayers surround the nucleus. And some information for you. For example, they have these pores pictured here. Not going to hold you responsible for that, but they are nuclear pores that allow certain substances into the membrane, into the nucleus. Okay? Okay. Now I need to move myself. There we go. So we talked about the nuclear envelope. We move to the other parts of the nucleus. The nucleoplasm, which is basically cytoplasm inside the nucleus. Cytocell, nucleus. Fluid inside the nuclear envelope. Very similar to cytoplasm. It's called nucleoplasm. That's all we're going to worry about for that. There's also this dense region here called the nucleolus. Okay, what I would know about the nucleolus, this is where ribosomes are made. Okay? We're not going to talk about ribosomes right now. We're going to get to them in chapter 11. But this is where they are made, inside the nucleolus. So remember I said know the function. So the nucleolus, its function is to make ribosomes. Okay, we're at the last part of the nucleus. We talked about nucleoplasm. We talked about the nuclear envelope. We talked about the nucleolus. We get to chromatin. So chromatin is the form of your DNA when your cells are not getting ready to divide. So I'm going to blow your mind here. Most of the time, your DNA is not in this form that we call a chromosome. Most of the time, your DNA is in this more diffuse, more spread out form of DNA called chromatin. And chromatin is defined as all the DNA and their associated proteins in the nucleus. So most of the time, your DNA is in this form, chromatin. The chromosome forms only when the cell is getting ready to divide. Why? Well, in cell division, your DNA needs to move. We're going to get to that in chapter 8 in more detail. But for now, just understand that most of the time your DNA is in this chromatin form. When your cell is getting ready to divide, the DNA is going to need to travel. It's going to have to move. So what happens is that chromatin condenses into chromosomes, a more condensed form. So we have 46 chromosomes that condense from chromatin. So I like to think of it this way. Let's say you're going on a trip and you need to move somewhere, you need to travel somewhere. So during cell division, the DNA travels. So that's why it condenses. When you travel, what do you do with your stuff? You stick it inside a suitcase and you close that suitcase so you can bring all your stuff with you. Wherever you go, you are condensing your stuff. That's what DNA is doing when it condenses into this chromosome form. You condense your stuff into suitcases so you can travel around, you can move around easier. DNA condenses so it can move around easier. So just imagine you moving around, you traveling without suitcases, you holding all your stuff. It's not going to be pretty. So DNA, when it's not getting ready to divide, is in this diffuse, spread out form called chromatin. When the cell's getting ready to divide, the DNA condenses into these forms that we know as chromosomes. Okay? And we have 46 of them. Again, we're going to talk about this in way more detail in chapter 8. We now move to our second category of organelles, the endomembrane system. And there are 4 major structures that are part of the endomembrane system, and I would know them. So, the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, and vesicles, which move things between the nuclear envelope, the endoplasmic reticulum, and the Golgi apparatus. I will talk about these three in way more detail over the next few slides. We've already talked about the nuclear envelope, so we're not going to talk about it again. Okay, so we talked about that already. We are going to focus on the endoplasmic reticulum, the Golgi apparatus, and vesicles. And what the endomembrane system is, they are a series of interacting organelles between the nucleus and the plasma membrane, okay, or cell membrane. Okay, so we are starting with the endoplasmic reticulum. There are two kinds of endoplasmic reticulum. Rough endoplasmic reticulum has ribosomes, those little bumps. Those are ribosomes. And if you remember, those ribosomes are constructed in the nucleolus. Okay, if you remember the nucleus. So, rough ER has ribosomes on the surface of it. And the function of rough ER is to modify proteins. And what I mean by that, it adds molecules to proteins. Okay, we're going to talk a little bit more about that when we talk about the making of proteins, which is in chapter 11. Okay, but the rough ER's function is to modify proteins. Now, the second type of endoplasmic reticulum, and you can shorten it as ER, the second type of endoplasmic reticulum is smooth endoplasmic reticulum, or smooth ER. Now, it's smooth because there are no ribosomes on smooth ER. That's why it's smooth. The function of smooth ER, the main function, and the one I'm going to hold you responsible for, is that is where lipids are made. There are other functions based on what kind of cell you're looking at, but the smooth endoplasmic reticulum's main function is to make lipids. And both ERs, the rough and the smooth, the proteins that get modified in the rough or the lipids that are made in the smooth, they will move to other parts of the cell via vesicles. So we're going to get to vesicles, but vesicles basically transport things between different parts of the endomembrane system and also different parts of the cell, or even out of the cell or into the cell. They are for transport. Okay? So, endoplasmic reticulum, to summarize, rough ER has ribosomes, modifies proteins. Smooth ER makes lipids, no ribosomes. Okay, we move to our third and fourth parts of the endomembrane system, the Golgi apparatus and vesicles. So the Golgi apparatus happens to be my favorite organelle. I know, everyone has a favorite organelle. Mine is the Golgi apparatus. Okay, maybe you don't have a favorite organelle. That's okay. That's okay. But my favorite is the Golgi apparatus. So what the Golgi apparatus does, it gets proteins that are modified in the rough ER. It gets lipids that are made in the smooth ER. They go to the Golgi apparatus for final touches. So that protein that's delivered to the Golgi apparatus, the final touches are put on. It is finished up in the Golgi apparatus. Lipids are finished up in the Golgi apparatus. The final touches are put on. What I mean by that, molecules are added. The final modifications are made in the Golgi apparatus. After the Golgi apparatus, those proteins, those lipids are done. They can move to a part of the cell where it's needed, or they might even move outside the cell to another cell in the body if they're needed. Okay? So how do they move? So they move via vesicles. These little circle things here, these are all vesicles. Vesicles. Vesicles can bring things into the cell. Vesicles can put things out of the cell. Vesicles can bring stuff from the rough endoplasmic reticulum to the Golgi apparatus. Vesicles can bring lipids from the smooth ER to the Golgi apparatus. So vesicles are there for transport. I like to think of vesicles as the UPS trucks. They deliver products either to different organelles, like the Golgi apparatus, or into the cell or out of the cell. Okay? So vesicles are there for transport. What the Golgi does is finish these off, and then they are sent away to where they are needed via vesicles. Okay, So we've talked about the 4 parts of the endomembrane system. We talked about the nuclear envelope. We talked about the rough ER. We talked about the smooth ER. We talked about the Golgi. We talked about the vesicle. Okay, I have bad news. I tried to insert this video into the lecture. I know I don't normally do that, but I love this song. It's a song about the Golgi apparatus. It's awesome, but unfortunately the audio wasn't great. Inserting clips from YouTube is not working so well, unfortunately. I tried everything. It didn't work. So I encourage you to watch this music video on your own. It has great images of the Golgi apparatus. And what I love about this song, it's biologically correct. It's amazing. He sings about the Golgi apparatus. It's kind of a happy song about the Golgi apparatus and what it does. He's a talented musician, and it's a little weird in the middle. What I love about this video too, it gets really dark. It's this happy song, and then right in the middle, there's a transition where he starts singing about, I don't know, lost love, a broken heart. I don't know. It seems like someone broke up with him. I think you should watch this video just for that transition. It's actually quite abrupt. Happy, 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 my heart's broken. It's crazy. So anyway, I encourage you to watch this song. I'm going to link this video on Canvas. Not mandatory. It's not going to be anything additional on the test about this song, anything like that. But it's a great song, and it's biologically correct. He does sing about the functions of the Golgi apparatus. And it's a song about the Golgi apparatus. Good God. So if you listen to that song, and if you didn't yet, that's okay, he will sing about lysosomes. Lysosomes. Lysosomes are specialized vesicles, and I would know them for a test. Okay, so if you remember, vesicles are used in the endomembrane system to move molecules from one place to another. They will take a modified protein from the rough ER and bring it to the Golgi apparatus. Or maybe that vesicle will take that modified protein from the rough ER and bring it outside of the cell or to a different part of the cell because it doesn't need to go to the Golgi apparatus. That protein is already done. Or maybe that vesicle takes a lipid produced in the smooth ER and brings it to the Golgi, or brings it outside the cell, or brings it to another place in the cell. Vesicles are the transport vehicles in the cell. They move things around. But there are some vesicles that are specialized for unique purposes. Lysosomes are an example of that. They are vesicles, but instead of moving things around, they are there to digest molecules or portions of the cell. What I mean by that, maybe a mitochondria that's no longer useful, or another organelle that is no longer useful, that has run out. Okay, so the lysosome would digest those portions of the cell that are no longer needed. What they have digestive enzymes. And more importantly, they are made in the Golgi, and I like them because he sings about them. Again, I like that song. It is biologically accurate. So, just to give you a little more detail on lysosomes, what they are, you have this molecule with digestive enzymes that is brought by a vesicle. Remember, transport. The transport vesicle brings those enzymes that are digestive enzymes to the Golgi apparatus. Then that vesicle emerges that instead of transporting, can now digest other things. So for example, it digests this molecule that is brought in this picture here. So that's lysosomes. That is their functions. They can digest molecules or portions of the cell, and they have digestive enzymes in them. And I'm going to link this video for you, which gives you a little more detail on lysosomes. I like this video. I will link it on Canvas. Another specialized vesicle that you do not need to know, it's in red, are vacuoles, pictured here. They can be found very prominently in plant cells. So this space can be used for waste disposal or storage. They are mainly found in plant cells, and they're much larger in plant cells than they are in animal cells. Some animal cells don't even have vacuoles, or they're very tiny. But that's why I'm not really holding you responsible for them. They are specialized vesicles for waste disposal or storage. Do not worry about them. Okay, here's a good example of a test question I may ask you on this upcoming lecture exam. Which of the following organelles is not part of the endomembrane system? Let's start here. Is the endoplasmic reticulum part of the endomembrane system? Yes, we know it is. Smooth ER, rough ER, part of the endomembrane system. Golgi apparatus, part of the endomembrane system. Vesicles, part of the endomembrane system. Nuclear envelope, part of the endomembrane system. The one choice that's not part of the endomembrane system is the nucleolus. So it's answer choice A. That is the correct answer. Nucleolus is found in the nucleus. And remember, its function is to make ribosomes. Correct answer is A. Okay. So we move to our next category of organelles, the energy-related organelles. We're going to talk about two organelles, one mitochondria, the second chloroplasts. So we're going to get into way more detail about mitochondria in chapter 7. So in chapter 7, hella more detail than we're going to get into than we are going to talk about now. Mitochondria are the ATP producers in the cell. If you remember ATP from last chapter, ATP is a nucleotide. Nucleotides are the monomers for nucleic acids, but some nucleotides have their own functions, like ATP. ATP is the energy-carrying molecule in our cells. It carries our energy. It has our energy. We make most of our ATP in this organelle called a mitochondria. Okay? Chloroplasts, they function in photosynthesis. Here's a picture of chloroplasts. Each of these kind of shapes here, that's a plant cell. Inside each plant cell, these green circles, those are chloroplasts. Plant cells have hella chloroplasts. Those chloroplasts function in photosynthesis. We're going to get into way more detail about chloroplasts in chapter 6. So, chloroplasts chapter 6, mitochondria chapter 7. For now, I would just simply know their basic functions. ATP producing mitochondria, chloroplasts function in photosynthesis. Another term that you should know is the theory of endosymbiosis. It is the idea that at one time, chloroplasts and mitochondria lived on their own as prokaryotic organisms. So endo means in, symbiosis means working together. It's the idea that at some point, mitochondria and chloroplasts lived on their own. They were prokaryotic organisms that got absorbed by a larger prokaryotic organism. And instead of being digested by that organism, it became part of it. Became part of that organism. So endo, in, symbiosis working together. It's the idea that chloroplasts and mitochondria once lived on their own, got enveloped by a larger prokaryotic cell, and instead of being digested by it, became part of that cell. And it's the idea of how the modern eukaryotic cell with chloroplasts and mitochondria came to be. Okay? Evidence for this? Chloroplasts and mitochondria have their own DNA and ribosomes. They don't use them anymore. They don't need them. But they still have them, giving the idea that at some point they did need them. They did use them. And at some point in their evolutionary history, chloroplasts and mitochondria had their own DNA and ribosomes, and they used it. So I have a video here that I will link to Canvas, which goes into a little more detail on that. So I think it's a really good video to watch. I'll link that to Canvas. Okay, so I'm going to go through these pretty quick because most of this is in red. Big thing about mitochondria, this is where we make our ATP. ATP is the energy-carrying molecule. Energy is in ATP. So where we make most of our ATP as eukaryotes is this mitochondria, this organelle. Again, we will go into way more detail in chapter 7. Here are some more details about this. We're going to talk about these more in chapter 7. For this chapter, makes ATP. Chloroplasts. So, again, we are going to go into way more detail in chapter 6 when we talk about photosynthesis. For now, all you need to know, the organelles of photosynthesis are chloroplasts. Here's what it looks like. We're going to talk about this again in chapter 6. Okay, going fast and furious through those organelles, we get to our last group of organelles, the cytoskeleton. So cyto means cell, skeleton means skeleton. You have a skeleton, okay? So it's the skeleton of the cell. And the function of the cytoskeleton is the same, has the same functions as our skeleton. It provides a structure, provides a shape, it allows for movement, Same thing with the cytoskeleton. So cyto means cell, skeleton, skeleton. It maintains the cell shape and structure, and it allows the cell as well as organelles within the cell to move. So just like our bones do not move themselves, our bones are moved by muscles. I am using muscles to move my bones. The same thing with cells. So motor proteins are inside the cytoskeleton, allowing for movement. So our skeleton is composed of bones, tendons, sinews, so on. The cytoskeleton is much, much smaller, so those structural components are also much, much smaller. The cytoskeleton can be composed of microtubules, actin filaments, intermediate filaments. They're pictured down here. Okay, you don't need to worry about what they look like for a test. I would just know the structural components of the cytoskeleton. Microtubules, actin filaments, and intermediate filaments. And again, they maintain the cell shape and they allow the cell and organelles to move. Okay, here's an example of how they allow the cell and organelles to move. So a sperm cell, for example, there's a little flagella at the end of that sperm cell. Here's a picture of cilia. You don't need to know too much about these. These tend to be composed of microtubules. These are structures found in cells which provide movement. That's about all you really need to know. Cilia, flagella, cell movement. Okay, I have a little video. You can watch unicellular organisms moving around with their cilia and flagella. We're almost done with this chapter. But first, we got to get outside of the cell. Oh my God, where do I put myself? Oh God, where do I go? I'll go here. I'll go here. There we go. I'll put myself right next to that plant cell. Yay! Okay, so let's move outside the cell. We talked about all the organelles inside the cell. We are now going to talk about outside of the cell. So plants, all plants, some fungi, some protists. I'll give you a hint. I don't like testing you on some. I like testing you on all. So plants have cell walls. Some fungi, some protists as well. Because they have cell walls, they need special structures to help them communicate with other cells. These special structures are called plasmodesmata. They are channels between two cells that have a cell wall. They are pictured here, close up over here. Imagine little tunnels between cells, allowing molecules to move between cells that have a cell wall. So animal cells don't got plasmodesmata. Plant cells do. Okay? We move to animal cells, the outside of animal cells. So plant cell junctions we just talked about, they are plasmodesmata. Okay? Animal cells have some junctions based on the type of cell. So there are three types in animal cells: adhesion junctions, gap junctions, tight junctions. These are connections between cells. So adhesion junctions, pictured here, are strong connections, but they allow for some flexibility. So adhesion junctions are found in your skin. So imagine, you can touch your skin, kind of pinch your skin. You can pinch your skin. You can do a lot of things to your skin and not break the seal. You're going to have to take a knife. Don't do it. Don't do it, for God's sakes. But you'd have to take a knife to break the seal or cut yourself open. But if you pinch your skin, you would have to pinch yourself hella hard to break that seal. So adhesion junctions found often in your skin. They are tight. They are very strong junctions, but they allow for flexibility. Just like your skin is flexible. It's not rigid. So gap junctions, pictured here and here. I'm sorry, pictured here and here. I'm sorry, I'm going crazy. Here's a gap junction. Here's a gap junction. What these junctions, they're between cells that communicate a lot with each other. So cells within your heart, for example, that need to coordinate the heart beating, they need to communicate quite a bit. They have a lot of gap junctions there. So that's what a gap junction is. Junctions or connections between cells that need to communicate quite a bit with each other. Now, here's our last junction we're going to talk about, tight junctions. So remember, adhesion junctions, they're strong, but they allow for flexibility. Tight junctions are very strong and not very flexible. You have a lot of tight junctions in your stomach to keep that stomach acid inside there. So when you need a really tight connection, a really strong bond with no flexibility, you get tight junctions. Okay, so an example in your stomach. So we are now at the end of this chapter. I just want to have a slide here kind of talking about how to prepare for the lecture exams, which is the hardest part of this course. So that study guide, I'm urging you, answer the questions in your study guide, memorize the answers to the questions in your study guide, and you will do very well on this upcoming lecture exam. You can use the book as a reference. You can talk to me if you have any questions, okay? If you don't understand something, please talk to me. I can meet with you online via Zoom. I can email you. We can talk on the phone. If you have any questions, do not hesitate to talk to me. That's what I'm here for. So I have some resources down here that the campus has. Given the situation we are in, some of these may not be open or have reduced hours or whatever. Again, if you have problems contacting any of these, let me know. I will help you. I will, I promise. Okay, so I'm just going to tell you the way I study, and whatever way works for you, do that. Okay, I have a certain way. It may not work for you. So maybe you use flashcards. Do it. Maybe you want to get into study groups. Do it. I tend to study alone. I tend to walk around. So luckily for me, my grandma was on The Price is Right and won a pool table. And she gave it to her son, my dad. So I grew up with a pool table, and I didn't play much pool, but I used it to study. I would walk around and talk to myself. That's how I study. Okay? That's what works for me. So whatever works for you. I do have to say something about biology. It is not easy. It is not easy. I don't think it's impossible to understand. I think it's a lot of material. So biology, I always say this about biology. Biology takes more time than you give it credit for. Okay? So there aren't any shortcuts. So put the time in. It's always a terrible feeling after an exam saying to yourself, wow, I could have studied more. I wish I studied more. It's much better to say after a test, wow, I studied too much. I studied too much. Oh well. So that's a better thing to say after the test. Put the time in. When I took my first biology class, I didn't do well. I actually withdrew. I took that same class the next semester, and I got an A. Not because my brain increased, not because I got smarter, but because I put the time in that was necessary. Biology takes time. Okay, so we're going to stop here. This is 4.2. I'm sorry if I went on too long, but we are now done with Chapter 4. I will see you in Chapter 5, Part 1.